everyone. Welcome to ITLS Academy, and this is Dr. Anita Srivastav. I'm going to talk about industrial microorganisms, and this lecture is in continuation to a previous lecture on industrial microorganisms. So here we are going to talk about the strain improvement techniques of industrial microorganisms. So, just going into the detail of this lecture, I would just brief about the microorganisms which are industrially important and why they are important to the industries. So, we know that microorganisms, they have been proved to be useful in industries. And why they are proved to be useful in industries? Because of their rapid growth, their mass cultivation, and the use of potentially economical substrate. So in previous lectures we have been talking about the substrates, about the some economical substrates that may reduce the cost of the industrial process. Then accessibility for genetic manipulation means microorganisms, industrially important microorganisms, they can be genetically manipulated. And with the advancement of genetic engineering, there have been unlimited possibilities that can improve their property for the industrial purposes at the industrial scale. So that means the accessibility for genetic manipulation has opened up with unlimited possibilities to improve their property for industrial scale. Then several processes, they were developed in last 2 decades, which involved the recombinant microorganisms and genetic engineering technology that improve the biotechnological application of the industrial strains. So previously or since last centuries, we have been knowing about the importance of the microorganisms in the formation of different kind of industrial products, like they have been used in the wine production, in basically beverages, pharmaceuticals. But previously the wild strains were used, and then with the advancement of this recombinant processes, there were some possibilities that the product yield may be improved, the productivity may be improved, that may reduce the cost of the process and make the process most significant or important. So the fermentation industries, actually the microorganisms which are used or preferred by the fermentation industries, they are known as the GRAS microorganisms. GRAS means which are generally regarded as safe for the manufacture of useful industrial product. That means they should be non-toxic, non-pathogen. So these kind of properties in the microorganisms which are seen, they are known as the generally regarded as safe or GRAS microorganisms. Then the commonly known microorganisms which are classified as GRAS. So they are categorized in bacteria, yeast and filamentous fungi. So when talking about the bacteria, the most commonly known bacteria which comes under this category or which are regarded as safe, these are Bacillus subtilis, Lactobacillus vulgaricus, Lactococcus lactis, Leuconostoc enos. And talking about the yeast, the microorganisms are Candida utilis, Cleveromyces maxianus, Cleveromyces lactis, Saccharomyces cerevisiae. Then among the fungi, Aspergillus niger, Aspergillus oryzae, Mucor zavanicus, Penicillium roqueforti. So these are the microorganisms which are normally classified as grass. Now, here we are talking about the strain improvement techniques. So basically, the factors, the factors that are to be considered for strain improvements. So this is the list of the factors that can be considered for the strain improvements. So first one is to reduce doubling time. Means the strain improvement, it can be used, it can have the reduced doubling time or the shorter generation time. So this factor can be seen for strain improvement. Then genetic stability. We should look for the genetic stability of the microorganisms. Non-toxicity to humans, they should be non-toxic. Large cell size for easy removal from the culture fluid, means during the process this is useful. Next is the potential to use cheaper or the economical substrate. So the microorganisms, the strain improvement can be done in this area where the microorganisms, they should follow certain pathways where they can use the economical substrate, they can use their carbon source or the energy source, which can lead to a good yield of the product. Then removal of the compounds that may interfere with downstream processing. As we know that downstream processing basically is responsible for the higher amount of cost production. So during downstream processing, there are many products which may interfere with this process. So the removal of those compounds is necessary. So at this area also this improvement can be done. Then obviously I'm talking about the productivity. So increased productivity should be the other target. Then to improve the use of carbon and nitrogen source which are used as their energy sources. Reduction of cultivation cost. Reduction of cultivation cost means we are talking about its growth, about its fermentation. So that should be reduced. Then affordable nutrition means the kind of nutrition they are going to take to further form the products. So that nutrition should be affordable, means it should be economical. Then microaerobic means they can process in the presence of a small amount of oxygen. And production of additional enzymes means the strain should be targeted to produce some additional enzymes. Compounds to inhibit contaminant microorganisms, and those compounds should be inhibited which can cause the contamination in the culture or the means basically the fermentation media. Now talking about or coming on the strategies of strain improvement. So basically the potential productivity of an organism, it is controlled by its genes, and therefore their genome can be or must be altered for the maximum production of enzymes. So the strain improvement techniques which are basically used are mutagenesis and genetic engineering. So these two are very important techniques that work in the improvement of the strain. Now talking about the mutagenesis. Mutagenesis means we are talking about the mutations. So in general, if we talk about the mutations, so any sudden heritable change in genetic material or character of an organism, this is known as mutation. And those individuals which are showing these changes, they are known as the mutants. And an individual which shows an altered phenotype due to mutation, they are known as variant, means mutants, variants. Then factors or agents that causes mutations, they are known as mutagens. Mutation which causes changes in base sequence of a gene, they are known as gene mutation or point mutation. So these were some terminology that was related to the mutation. So as I said, the mutations, they result from a physical change to the DNA of a cell, such as deletion, insertion, duplication, inversion, and translocation of a DNA piece. A change in the number of copies of an entire gene or chromosomes also result in mutation. And this mutagenesis, it is a process of treatment that is given to the microorganisms which will cause an improvement in their genotype and phenotype performances. So if we divide this mutagenesis, it is of three types, that is spontaneous mutation, direct mutation, and induced mutation. Now talking about the types of mutation. Talking about the spontaneous mutation. These spontaneous mutations, they are known as they are known as natural mutation, and a mutation that arises naturally and not as a result of exposure to mutagens. Their rates are very slow. The spontaneous mutations, they occur from abnormalities in cellular or biological processes, that is due to the errors in the normal functioning of cellular enzymes and errors in DNA replications. So these are the spontaneous mutations. And if we talk about the spontaneous mutations rate in eukaryotes, it is between 10 to the power of minus 4 to 10 to the power of minus 6 per gene per generation, whereas in bacteria and phages, they are 10 to the power of minus 5 to 10 to the power of minus 7 per gene per generation. Now coming to the induced mutation. A induced mutation, it is produced by the treatment with a physical or chemical agents, and it affects the DNA molecules of our living organisms. Then the rate of mutation, it can be increased by various factors and agents, and they are known as mutagens. Now, physical mutagens, they include the UV rays, which are non-ionizing, whereas when we talk about the physical mutagens like gamma and X-rays, they are ionizing radiations. So, depending on the ionizing feature, physical mutagens are categorized into the ultraviolet, gamma and X-rays. Then coming to the chemical mutagens, they are compounds such as ethane methane sulfonate, that is EMS, and we have nitrosomethyl guanidine, that is known as the NTG, and we have nitrous acid and acridine mustards. These comes under the chemical mutagens. Now talking about the point mutation or substitution mutation of a nucleotide. So all types of point mutations, they occur spontaneously during SG1G2 phases of the cell cycle or by the movement of the transposons. The and the point mutations, they are actually when there is a change in one base. For example, we can see here, this is a point or the substitution nucleotide. So here we can see that at this position means there is a change, that is AATA, and here we can see that there's a change at this position and this adenine is being replaced by the thymidine. Whereas in the deletion of a nucleotide, talking about the point point mutation, means the point mutation and this deletion mutation, it will be talked under the frame shift mutation. So here we can see that the deletion of a nucleotide means we can clearly see that this adenine at this in this base pair at this point this A is being deleted. So this is an example of the deletion of a nucleotide. Now some specific diseases that are caused by point mutations. Those are cystic fibrosis. It is a defect in the cystic fibrosis transmembrane conductance regulator gene that causes cystic fibrosis. Then neurofibromatosis, sickle cell anemia. It is also an example of point mutation in the beta globulin chain of hemoglobin, where glutamic acid is replaced by valine in the sixth position. Another example is the color blindness. Now talking about the example of deletion. So cystic fibrosis. Cystic fibrosis, it is a hereditary disorder and it is characterized by lung congestion and infection and malabsorption of nutrients by the pancreas. So these are few examples of the point mutation as well as deletion. Now talking to the addition. Now talking to the addition of a nucleotide. Basically, frame shift mutations, they occur when a protein is drastically altered because of insertion or deletion. So we can say that this insertion means addition or deletion. There are the examples of the frame shift mutation. So here in addition we can see that AAT, and here A, this is A, it is being added G. So this A and G, so a G is added in between T and A. This is a addition of a nucleotide. Then substitution of a nucleotide, we can see that at this position this cytosine is being replaced by the thymine. So we can say that the example of this frame shift mutation is Tay-Sachs disease, where they are increased to the susceptibility at certain cancers and classes of familial hypercholesterolemia. And then this is the basically this is addition and substitution of nucleotide. So here we talked about the spontaneous mutation, about the induced mutation, and point as well as the frame shift mutation. So now coming to the genetic engineering of microorganisms, the development of recombinant DNA technology and methods of cell fusion such as hybridoma formation and monoclonal antibodies production, they had a major impact on industrial microbiology. Then the recombinant DNA technology, actually it uses genetic engineering to introduce genes into microorganisms and other cells. Now coming to the recombinant technology, insulin, vitamins, amino acids and enzymes, they are the products of recombinant or genetic engineered microbes, and they have been playing a very useful role for the benefit of the humankind. Now currently the genetic modified organisms, they are have wide applications in human health, agriculture and bioremediation, and in the industries such as food, paper and textiles. So genetic engineering, they offers sufficient supplies of desired and cheaper products. Now the molecular tools for genetic engineering which are used by microorganisms, that is the gene transfer method to deliver the selected genes or the into the desired host. Then the cloning vector, promoters to control the expression of desired genes. And the fourth is selectable marker genes to identify recombinant microorganisms. So talking about the gene transfer method, there are three methods. We'll be talking about the transduction, transformation and the conjugation. So talking about the transduction it is defined as the transfer of bacterial DNA from one bacterial cell to another by means of a viral infection, that is a bacteriophage. Then there are two types of transductions which are known, means generalized transduction and the specialized transduction. In generalized transduction, host DNA is taken from any part of the host genetic apparatus and it is integrated into the virus DNA. And then talking about the specialized transduction, in specialized transduction, it occurs only in some phages and DNA from a specific region of the host, it is integrated into the viral DNA, which replaces some of the virus DNA. Means in generalized, DNA can be taken from any part of the host cell, but here in case of specialized transduction, DNA is taken from a specific region. So here in this figure we can see that this is the bacterial DNA and this is the phage DNA. So first one here this way we will see that this is a phage protein code bacterial chromosome and this DNA phage this is entering over here and this is the donor cell. So the phage it infects the donor bacterial cell. Then in the second step here we can see that the phage DNA and proteins they are made and the bacterial chromosomes is broken down into small pieces. Here we can see that occasionally during the phage assembly the pieces of bacterial DNA they are packaged in a phage capsid and then the donor cell lyses and release the phage particles containing the bacterial DNA. So this is the recipient cell and here the phage carrying the bacterial cell, it infects a new host cell or the recipient cell. So we can see that the recombination can occur producing a recombinant cell with a genotype that is different from both the donor and the recipient cell. So this is donor bacterial DNA and this is the recipient bacterial DNA. Now this is as I was talking about the generalized transduction. So this here we can see that the phage infects a bacterial cell, then destruction of bacterial DNA, synthesis of viral DNA and proteins, and here we can see that the virus capsid synthesis and assembly takes place, then lysis of the bacterial cell, phage release and infection of new cell. So basically a recombinant is being formed. Then talking about the conjugation, the second type of gene transfer method. So basically the conjugation it is defined as a process where the transfer of DNA it directly from one bacterial cell to another by a mechanism which involves cell to cell contact. Means here we can see there are two cells. This is a mobile plasmid and this is the pilus, and through this pilus we can see this is the recipient. So it is DNA is transferred in a nucleus resistant form which differentiates it from transformation and without the aid of bacteriophage which distinguishes it from transduction. So this is the process that how the conjugation is taking place. Now talking about the transformation. So certain microorganisms they are able to take up the naked DNA that is present in the surrounding medium, and this process it is called as transformation, and this gene transfer process is actually it is limited to strains that are naturally competent. So here on this figure we can see that this is the nucleide and this uptake of DNA is taking place. So if the DNA integration by non-reciprocal recombinant so stable transformation is taking place, whereas when the degradation takes place means it is an unsuccessful transformation, means when this DNA is not these DNA fragments they are not taken up by the cell, it is basically the unsuccessful transformation. So some therapeutic products that are made by the genetic engineering and they are erythropoietin factors, means talking about the blood proteins, they are erythropoietin factors that is 7, 8 and 9, tissue plasminogen and activators urokinase, and these are the functions which are given. And talking about the human hormones we can see epidermal growth hormone, follicle FSH follicular stimulating hormone which is used for the treatment of reproductive disorders, insulin for the treatment of diabetes, nerve growth factors, relaxin, somatotropin, and somatotropin basically it is used for the treatment of some growth abnormalities, And talking about the immune modulators, we have alpha interferon, beta interferon, we have the CSF, that is the colony stimulating factor, lysozyme, tumor necrosis. Then replacement enzymes like glucocerebrosidase, it is used in the treatment of Gaucher disease, that is an inheritable neurological disease. And some therapeutic enzymes, they are the human DNase 1 and alginate lyase. And the human DNase 1, it is used for the treatment of cystic fibrosis, whereas alginate lyase, it is used in the treatment of cystic fibrosis. So there are a number of therapeutic products which are made by the genetic engineering. So basically, it is all talking about the strain improvement techniques of the industrial microorganisms. Thank you.